Hi, welcome to a Physionic detailed study analysis. Today we're going to be going over glucagon-like peptide 1 agonists and their effects on weight loss, as well as metabolism and a few other metrics as well as understanding the mechanisms of how they actually function and why they're so effective, or supposedly so effective. We'll find out. So we're going to be going over 11 different studies to come to some form of conclusion on their effectiveness. And this is going to be a deep dive, so we're going to be going into a lot of data and mechanisms and all of that kind of stuff. So if you're interested in a shortened version, then just jump to the conclusion sections where you'll get a condensed summary of what we've been over. However, you're going to miss out on a lot of the detail, so I would encourage that you stick it out. That said, if I make any sort of mistakes or need to make any sort of amendments after the fact, I will put a amendment that will be linked to this content, so look out for that as well. All right, if you're not familiar with who I am, my name is Nicholas Verhoeven. I'm a PhD candidate in molecular medicine, and this is what I do. I take studies, aggregate them together, and then keep them open so that you can actually understand and look at the data for yourself and come to conclusions that may be different from my conclusions. All right, so without further ado, let's jump into this. All right, so the topics that we're going to be covering are obviously GLP-1A's mechanisms of action, and then we're going to go over the weight loss effects, so looking at all the data on the weight loss, and then also the metabolism effects. Now, if you're part of the Physionic Insiders, you'll also get access to which specific GLP-1 agonist seems to be the most effective. I know a lot of the data has been focused on semaglutide, but there's actually one that seems to be far superior to semaglutide. So if you're interested in that, most effective doses without side effects. So we'll go over a little bit on side effects as well. So I'll describe the effective doses that minimize side effects, and then also comparison study, a meta-analysis on GLP-1 agonists versus weight loss surgery, looking at bariatric surgery, which I found really interesting. So I thought I'd add that as well. If you're interested in having access to the full version of this video that you're watching, then just hop on over to the Physionic Insiders, join there, and then you'll have access to this plus a whole library of other videos, and you'll have a written summary of this video as well and a bunch of other stuff as well. So I'm not going to go into all the different benefits at the moment, but there's a tremendous amount. So I'd encourage that you join, and there's a link that you can join. And otherwise, let's go ahead and jump into this. So the mechanisms of action. For this, we're going to be relying on this study, which is a scientific review, and it's study 226 of my library of studies that I've gone over. It's unknown funding, and there are some conflicts of interest, meaning that the researchers have some sort of tie to a pharmaceutical or something of that nature. You can find more details on all that in the notes that I have attached to this content for you, as well as the actual study and the information, the DOI numbers, and all that good stuff. All right, so let's explain how glucagon-like peptide works. So glucagon-like peptide is a molecule that's secreted from primarily your L cells within your intestines. So here we've got the intestinal brush border over here, and all these intestinal epithelial cells line or make up your actual intestine. So if we were to zoom out and actually look at a piece of your intestine, it would look like this. Then if we zoom in, it would look something like this. And within this epithelium, you have different types of cells. Like in the stomach, you might have like goblet cells. Well, in here we have L cells, and L cells are the main one responsible for the production of GLP. Now, the activation of GLP, glucagon-like peptide, we're not talking about glucagon-like peptide 1 agonists yet. We'll get to that, but they work through an extremely similar mechanism to what we're going to be explaining here for glucagon-like peptide. So they're produced by the L cells, and the reason that they get produced is when we get a sensation of nutrients that are going through the intestines. So if nutrient load increases in the intestines, that gets sensed by the cells, by these L cells, and then they start producing more of this glucagon-like peptide, which they then dump into the circulation. And once it's in the circulation, it has a number of different effects, but we'll get to that in just a second. So glucagon-like peptide is actually very similar to glucagon, which is another hormone that has tremendous effects throughout our metabolism and our physiology. And I wanted to explain this real quick, that if you look at an L cell membrane, so if we're inside of a single L cell, so if I can go back here, we're inside one of these cells. We've got our membrane here. We've got the inside of the cell, the cytosol, and we've got the outside of the cell right here. We've got the nucleus, which is where our genes are kept. And what I wanted to point out here is the way that the genes are expressed. So I don't actually go into this detail too much, but there's a lot of complexity. Of course, there's a lot of complexity when you're talking about the genome and if you're talking about molecular biology. But you've got these genes that can be extremely similar to other hormones. So they can actually be basically the same gene, but the way that they are expressed by the cell can be slightly different. So what do I mean by that? So the genome can be, let's say, gene A contains the information for glucagon as well as the information for glucagon-like peptide, which are two distinct molecules and have distinct effects on the body, but they arise from the same gene. How does that happen? Well, the cell will have its gene reading machinery come in and attach to the gene, and will then read that gene into a mRNA. So the gene here in this situation is made up of 3 different sections. So it could read maybe just the green section, or it could read just the yellow section, or just this pink section, purple section, or it can read 2 of them at once, or it can read the green, then skip over the yellow, and then read or express the purple in combination with the green. There's a number of different ways that the cell can read particular genes. Now, ultimately, this leads to the production of what's known as an mRNA, a messenger RNA, which is what the gene is then is actually transcribed to. So it gets transcribed from a gene to a mRNA. And then the mRNA can be further acted upon by what's known as the spliceosome. So the spliceosome will come in, and which is another piece of gene editing or gene regulatory machinery within our cells, and will then cut at particular sections of the mRNA and get rid of certain sections. So even if the gene were to make an mRNA that included the green, the yellow, and the purple, the spliceosome can come in and cut away the yellow and the green and just leave this mRNA. So maybe the whole gene being read without the spliceosome having an effect would read for glucagon and produce the glucagon protein. Or if the spliceosome comes in and cuts so that it only has this purple mRNA, then that leads to the GLP-1. I just wanted to point this out because it's something that I find really fascinating, and it also displays the complexity of how genetics works and how epigenetics works and how molecular biology works. It's far less straightforward than what you usually get taught. I'll just put it that way. And even what I'm explaining here is still a dumbed down version of what actually really happens. There's many different molecules that are going in and out of these different functions, and they have specific effects at specific times. So there's a lot of complexity, a lot of regulation that goes into this. So anyway, the point is that we get what's called an alternative splicing of the mRNA into the GLP. And then the GLP mRNA gets out of the nucleus, and then it undergoes protein synthesis where ribosomes will then attach and then stitch all these tRNA with amino acids to it, create a polypeptide chain. And then from there, this chain of amino acids then get folded together into the actual GLP-1 molecule, and then it gets excreted out of the cell. So that's the way that GLP-1 actually gets produced. Now, once it gets produced and it's in the bloodstream, like we see here, then it can go through circulation, and certain GLP-1s, or I guess in general GLP-1, can actually affect our brain. And that's actually how the proposed mechanism of how it actually directly affects our weight loss. So GLP-1 will then bind to a region in the brain called the hypothalamus. So if we were to zoom into the hypothalamus, what would we see? We would see 2 different groupings of neurons, which are brain cells. Now, normally, under normal conditions, let's take GLP-1 out of the equation for a second, and let's look at POMC neurons. So if POMC neurons are activated, what they do is they actually inhibit food intake. So they secrete molecules and activate or deactivate, I should say, inhibit other neurons that are responsible for our want for food, okay? So they increase our satiety. The activation of POMC neurons typically increases our satiety directly as well as indirectly by affecting NPY, neuropeptide Y, and AGRP, agouti-related protein neurons. So this is the you can think of it like the opposite of the POMC neurons. These neurons, if they are activated, they will increase food intake. So then they will have a direct effect on other parts of the brain and activate those sections to be less satisfied and want more food. Okay, so POMC neurons can have an indirect effect by blocking these NPY-AGRP neurons, and therefore then they're blocked from food intake, or they can directly have an effect on food intake by inhibiting the downstream neurons that both of these groupings of neurons have an effect on. Okay, so that's how it would normally work. Now GLP, if you see rises in GLP-1, then GLP-1 will bind to these POMC neurons and will then directly affect and then activate the POMC neurons to then reduce the food intake, again, by deactivating downstream neurons, and as well as deactivating the NP1-AGRP neurons. Now, additionally to that, GLP can directly bind to the NPY-AGRP neurons and can inhibit them so that they don't activate downstream neurons. So that's the general idea of how GLP-1 works. Now, there's a problem though. And why, because then why not just inject ourselves or just consume GLP, right? It's the thing that's produced by our body. So why not just use that? Well, the reason why is because we've got these peptidases, these DPP-4 proteins, a specific isoform, particular type of DPP protein, that will attach to GLP-1 and break it up. And it actually breaks it up super fast, within minutes, if I remember correctly. So these DPP proteins will recognize, not that they specifically consciously say, oh, there's a GLP-1, but their molecular structure allows them to attach to GLP-1 molecules and thereby degrade them, like we see here. Okay, so the solution then is, and this is the only slide I have on GLP-1 agonists, is that GLP-1 agonists will act like GLP-1, but are far less sensitive to DPP. So the DPP now no longer can affect GLP-1 agonists nearly as much as the normal GLP-1 that's secreted by our body. So they're structurally very, very similar in that they can still have all of these effects, but they are insensitive to DPP, or far, far less sensitive, I should say. They can still get degraded, and they do eventually get degraded, but it can take days as opposed to minutes, or it could take up to hours, days, or even weeks before they get degraded, unlike the normal GLP-1 that we see here. All right, so we've been over the mechanisms. Now we're going to go over the actual data itself. So look at how GLP-1 agonists affect weight loss. For that, we're going to rely on a few different studies, but we're going to start out with study 217, which is publicly funded, so has no ties to any sort of industry or anything like that. And this is a meta-analysis. So they looked at hundreds of different studies, ended up excluding many different studies because of various reasons, like some of the studies were reviews or commentary sections. They were other meta-analyses. They were abstracts, which means that they didn't actually come with the data themselves. They were non-randomized controlled trials because all these studies were randomized controlled trials that they wanted to include. They're non-English language. There's a number of different reasons, but ultimately they ended up with 8 studies that they could include in the meta-analysis. And then when we look at the bias assessment, so the way it's broken up is in 3 sections. There's green, which indicates low risk of bias. Red, which means high risk of bias. That doesn't mean that there actually is bias, but it means that there's a high risk. And yellow, which indicates that there is, it's unclear what the risk of bias is. And they go through a number of standardized questions that they have to answer, and therefore then have a specific number that they get, and then they relate that number to, is it high risk or low risk of bias in these different conditions. So, for example, they would ask, is it double blind? Is it randomized? Things of that nature would all be taken into consideration here. Now, I will say that this is an incredibly good risk of bias assessment. This would indicate that there's very, across the analysis, the entire analysis, there's very low risk of bias. So this is really, really fantastic to see. All right, so let's look at the data itself. Now, here we've got to walk you through here. I'm going to walk you through this once, and then I'm going to blast through the other data so that I'm not constantly repeating myself. So on this side right here, these are all the studies that were included. Now you'll see O'Neill, for example, is listed like 6 or 7 times. The reason for that is because they're using different concentrations of the GLP-1 agonist. And so the reason why they can do this is because they have separate subjects and separate time points that they do to get this data that they're getting. So that's why they can include it. Now, it would be a different story if they used the same subjects and just used different measures and then included that in the analysis. That would not be allowed. That would be against statistical protocol. So, but in this case, it's fine because, again, they're looking at different groups of individuals. Now, with all these, and then you've got all these other studies as well. So we've got the studies listed here. Then we've got the effect of semaglutide. So they're looking at a specific GLP-1 agonist in this situation versus placebo. And the placebo is then supposedly an inert subject, a substance of some sort. So it's not going to be, something like metformin or some other drug or anything like that. It's not supposed to be a bioactive ingredient. It's supposed to be something that is supposed to mask some sort of an effect. So semaglutide, here we've got the mean. So here are all the average results that they experience. So if you see a minus, that means that they lost weight. And standard deviation gives you the variance of the effect. So some people lost more weight, some people lost less weight. It just gives you the variance, the overall spread of the data. And the total is the number of participants that were found in each group. So semaglutide for, let's say, the Friedrichsen study was 36 individuals, or the O'Neill, this first O'Neill one was 103 participants, the second was 102 participants, et cetera, et cetera. And then the same thing for the placebo. You got the means for each study, standard deviations, and the total. Then they've got the weight here, which indicates the amount of power, the amount of pull. I think that's the more appropriate way to describe it. The amount of pull that each study has on the entirety of the analysis. So how much does this O'Neill 2018 study pull or have an effect on the overall analysis of the study? And these are extremely even. This is, I can't stress this enough. This is really great to see. I'm loving the fact that the risk of bias is very, very low. And I'm loving the fact that the weight of the analysis is incredibly evenly distributed. So this is fantastic. Then here they've got the mean difference, which is typically the comparison of the semaglutide mean versus the placebo mean. So they're looking at the mean difference, and this is what it's quantified to on the left. And then they've got the confidence interval, which is where the true mean could actually lie. So while the statistics are going to indicate an estimated mean, which is in this case for the Friedrichsen study, I have to pull out some German here, minus 9.41. So that's the best estimate of the actual result comparing semaglutide versus placebo. And then, but the true mean, what is actually the true answer may actually fall anywhere between minus 11.51 and minus 7.31. So somewhere in there. But the point is, all of these numbers are negative. All of these numbers indicate that it's in favor of semaglutide, okay? So that's how to read that. And we see that we can see all these numbers are actually represented by these green dots, which is the minus 9.41, and the lines going through are the confidence intervals. And they're using a random effects model. You know what? I'm not even going to go into that. But the point, I'll go ahead and say that it is the appropriate statistical test to use in this circumstance. Okay, so that's how to read it, and what we can, and it's really easy to read by just looking at the values for looking at the actual diagrams here. So each one of these studies is indicated here, and you can see that if it's on 0, if all the studies were on 0, that would indicate there's no benefit one way or another. So there's no advantage one way or another in terms of weight loss. If it were to the right, so let's say if it were at 5 or 10 or something along those lines, then it would be in favor of the control or in favor of placebo, which would indicate that semaglutide does not have a weight reduction effect and may actually lead to weight gain by comparison to the placebo. But there would be some nuance there. Now, what we actually see, though, is that everything is to the left. Everything is firmly planted to the left. This is excellent consistency in terms of the overall effect. Now, the overall effect is also represented. You can look at each one of these studies individually, or it's also summarized in this black diamond. The black diamond is firmly, squarely planted towards the left, which would indicate that this analysis indicates a reduction in body weight. So this is a relative body weight. Now, when we look at for the test of the overall effect, we see a z-score of 11.27, which is massive if you're into statistics. The p-value is well below the cutoff point, the typical cutoff point of 0.05. It's way below. So there is a statistically significant effect. Now, there's high amounts of heterogeneity as well, statistically significant heterogeneity, and the heterogeneity is extremely high, and that's actually why they used a random effects model. But the point is that I feel extremely confident in saying yes, GLP-1 agonists across the board extremely consistently show weight loss effect. Okay, here I wanted to point this out. You'll see that there's a big jump. So I go at figure 4 and then I go to figure 7. The reason why I skip a lot of the other data is because every single other study or every other data analysis in this meta-analysis indicates the exact same effect. So they're looking at like waist circumference or body fat, things like that, which are other proxy measures of essentially the same thing, and they all consistently show this effect. So instead of boring us and just repeating the same thing over and over, I went ahead and skipped it. But of course, you can open up the study for yourself. And now that you understand a little bit on how to read it, you can see it for yourself. But this one was a little bit out there in terms of understanding it. And this one even threw me for a loop, honestly, because I thought this was really poorly explained. And the way to read it is extremely similar. So we're still doing semaglutide versus placebo, et cetera, but you'll notice that everything is on the right side and is in favor of control. Now, this was incredibly confusing for me. And when you read the actual analysis, the actual written section, they actually indicate that this is an indication that that fewer people were able to achieve at least 5% weight loss in the control condition. That's how to read this. It's not saying that the semaglutide condition over here had worse results. It actually indicates that it had better results in getting more people to achieve at least 5% weight loss. And I wanted to specifically point this out because it just looks very confusing, especially by the fact that what I explained earlier. But again, this is a consistent result across the board. Okay, and then another one that's pretty confusing is the adverse events. So we're talking about the side effects that people experienced. So the side effects, I'll go ahead and tell you about those and the actual side effects themselves in just a little bit. Let me just explain what this is. What they're looking at here is, again, the control versus the semaglutide. And this is, again, extremely confusing how they displayed it. But if you read what they wrote, they ended up indicating that this test for overall effect indicates that there's an effect against semaglutide. In this situation, we're talking about it's better to be on the placebo than it is to be on the semaglutide, meaning that there are more adverse events that occur with semaglutide compared to placebo. This is not clear at all. The reason why I say that is because if I were to read this, just looking at this, I would come to the opposite conclusion, because the number of events, which I would interpret as side effects, is let's say 93 here, and it's 107 under the placebo, which would indicate that there's more in the placebo. However, again, the way the researchers have actually explicitly written it, they say that there's more side effects with the semaglutide condition. This is incredibly confusing. That's why I'm making a specific point to show it to you, because it is just that confusing. Now, the actual side effects themselves. There were two main ones, and I'd like to address a third. The first one is nausea, and the second one is diarrhea. Those two were the main side effects experienced. Now, some people experience stomach discomfort as well. So just in that general realm. So it's not anything too extreme. It's not like heart attacks or anything like that, but it is a side effect. These are side effects to still keep in mind. Now, the other thing to mention is that some people have worried about muscle loss as one potential problem with semaglutide or just GLP-1 agonists in general. I tried to look for data on that. I have found no data indicating that there's muscle loss. That said, that does not mean that it doesn't happen. Just to be 100% clear, just because I didn't find any data doesn't mean that it doesn't happen. It just means that it potentially doesn't exist at this point. And once that data comes out, I'll of course report on it. But as it stands, there are more side effects with semaglutide, but they're not super serious as we understand them currently based off this literature. All right, so the next step is to look at study 218, which is again public funding again. And in this one, we're going to get a little bit more granularity on the effects semaglutide has. It might be important for you to actually know which GLP-1 agonist is the best out there. And there's one that's actually covered in the extended version of this video. If you're interested in accessing that, along with all kinds of other videos, deep dives with summaries, including this one getting a summary as well, then certainly consider joining the Physionic Insiders. I'll put a link, a ticker in this video, as well as post the link down below. It really is the bee's knees of premium content, if I may say so myself. With that, let's get back to the video. Here we've got the number of studies that they included, which is 41 in total. And what I like about this analysis is that, well, I don't really like the way that they display it, but let's put that aside for a second. But the reason why I like this is because of all the studies that they include, they break it up by the types of GLP-1 agonists, the types of controls. So the studies may compare. So the reason why the previous study had only 8 studies and this one has like 40 something, did I say that right? 41 studies is because some of the studies that this analysis included were GLP-1 agonists versus lifestyle modification versus metformin versus placebo. So a number of other versus comparators, as opposed to the previous study only was limiting it to just the placebo. So when we look at those values, so they break it up according to how does GLP-1 agonists have an effect, or is it useful if you compare it against a placebo, if you compare it against lifestyle modification, metformin, etc., as well as by treatment duration. So if it's like short, if it's like days or weeks versus months, many months, or sample size, if there's a smaller group of individuals or a larger group of individuals. Study quality, is it high risk of bias, low risk of bias? And then the different types of GLP-1 agonists. So this is looking at mean differences, and the reason why I'm pointing that out is going to be important. So what we've looked at is mean differences means that comparing the average result of semaglutide or liraglutide or any one of these other ones, which I refuse to pronounce because they're tough. So comparing the mean effect or the average effect versus whatever is being compared against, the placebo, lifestyle modification, metformin. So that's where we're comparing the two averages against one another. So I'm going to focus on this column, so the body weight change and the total body fat change. And then you can see by the color the amount of certainty about the evidence. The certainty of the evidence is dependent on the confidence interval that I explained earlier. So if you have some of the results are in the positive, so meaning that some studies show, okay, yeah, there's an effect of placebo. So placebo is doing better. And in other studies, semaglutide is doing better, or liraglutide, or whatever it is that they're using is doing better. It's kind of all over the place. That's going to extend the confidence interval, and therefore the certainty is going to decrease quite a lot. And that's why the ones that are in red show low levels of certainty. Okay. So that all explained. Body weight changes. So in total, all the studies combined, there seems to be about a minus 5.31 kilogram drop in body weight when you compare all the studies together. That's including the ones, placebo, lifestyle, et cetera, all together. Total body fat change, there is a drop in body fat as well, but it's much lower. So it's 0.32. Now that doesn't, and these are percentages. So we're looking at percentage for body fat. So don't think that minus 5.31 kilograms, but they only lost 0.32 kilograms of fat. That is not how to read that. It is minus 5.31 kilograms of weight, and they lost minus 0.32% body fat off their total body fat. So it's probably going to be maybe a few kilograms of body fat. So just to be clear, because that would really trip someone up. So this would indicate that there is an effect of the GLP-1 agonist, which is what we saw with the previous analysis as well. Now, does it depend on the type of GLP-1 agonist? Here we have all the data for the body weight. All of these are consistently lower. So semaglutide has the greatest effect based on these metrics. Now, if you're part of the Physionic Insiders, I'm going to go into another one, or maybe another two, but definitely another one that is more effective than semaglutide. But as it stands, based on these comparisons for body weight, semaglutide seems to be the most effective. Now, the effect for the total body fat percentage, there's a little bit of discrepancy there. So one of them, and they don't have information for two of them. One of them, it does seem to have a reducing effect, and the other one, it seems to be about moderate, maybe even a slight increase in an effect. But there's also a much lower effect on body weight when using exenatide. So exenatide may not be the best option when coming to, or really isn't, for sure isn't the best option when coming to body weight loss. The types of control. So when comparing against placebo, there seems to be an effect when comparing against lifestyle modification, which is really important. Also, it seems to be better with the GLP-1 agonist. When comparing against metformin, the effect size is a little bit lower and also seems to have a benefit. So in favor of these GLP-1 agonists. And that seems to be the case across the board for when looking at body fat percentage as well. Treatment duration, again, we're seeing effects across the board. The short doesn't seem to have, really any effect on body fat percentage, but again, the uncertainty is kind of all over the place. The confidence intervals, I should say. Sample size, again, we're still seeing effects. If it's a small sample size or a larger sample size, we tend to have a lot more certainty and a lot greater effect when looking at larger sample size. The same for body fat percentage. Study quality doesn't seem to matter too much. Again, the point is we're seeing some consistency on any one of these GLP-1 agonists, maybe not quite as much for the exenatide, but for the others, for sure, we see pretty significant effects on body weight and body fat percentage. Now, this is, if you didn't see, we've switched to another slide, so this is different. This is looking at the exact same thing, but and I wanted to report this because the previous data, this data that we just went over, compared to this data, the previous data was looking at mean difference. So just comparing the average versus the average, again, GLP-1 agonist versus placebo, for example. This one is looking at the standardized mean difference, which is essentially a meta-analysis technique to take, there's multiple ways of measuring, for example, body weight or body size in general, or whatever it might be. So as a result, they standardize it. The meta-analysis researchers of this analysis that we're looking at standardize, essentially come up with an algorithm or a calculation to standardize all of the results that they have from all the raw data from each one of these studies, so that it can be a more equal playing field, if that makes some sense. And again, we don't really see any sort of huge differences across the board. It's going to be roughly the same in both situations. So if you're looking at mean difference or standardized mean difference. All right, the final one is to look at GLP-1 agonists on metabolism. So we're going to be leaning on study 225, which is unknown funding. I couldn't find the funding source for this, unfortunately. And here they include 24 studies in the overall analysis. However, when you look at the actual analysis itself, the meta-analysis itself, they only include 3 studies. Now, admittedly, I was very confused by this. So I looked at this a little bit deeper, and I found out that some of the studies were excluded. So some of these, the 24 studies here were excluded because they only lasted 1 day or just a few days, which isn't enough, or they didn't report on energy expenditure the way that the researchers of this analysis wanted them to. Now, on the other hand, however, there were other studies that I looked into that did report everything and seemed to follow all the criteria that these researchers had outlined. And for some reason, they just didn't include them in the analysis. So I'm very confused by this. And they only focus on one. They only focus on one of the GLP-1 agonists, so liraglutide. So they don't look at multiple other GLP-1 agonists. So I think this data, I'm not saying anything's fishy is going on, but I'm just saying that I don't understand. They don't explain why they eliminated certain studies that I think should have been included in the analysis. So I'll just go ahead and say that we have extremely limited data to make any sort of conclusions here. We've got 3 studies that are included, and there's no statistical significance here, but we only have 2 studies that are really large enough. And they seem to sort of lean in favor of placebo, as in like you get a lowering of metabolism with the liraglutide, but technically it's non-statistically significant, so we can't say that. I just don't think that we have enough data here to be 100% certain about, or even 90% certain that GLP-1 agonists have no effect on metabolism. As it stands, based on these studies, it does not seem to have an effect on metabolism, but I would like to see a lot more on this to be 100% certain. Okay, so the midpoint conclusion here is that GLP-1 antagonists, or agonists, I should say agonists here, are highly effective at causing weight loss with a strong body of evidence behind them. There's serious consistency across the board. There's some minor side effects reported from like nausea and diarrhea, and I had mentioned about muscle loss. There's no data up to this point, but as more data comes out, hopefully I'll be able to cover that. And that may also be avoided at lower doses. So the next questions that we have, if you're interested in having the extended version of this, is to look at which GLP-1 agonist is actually the best of all of them. One of them that I haven't even reported on up to this point. What's the effective dose that leads to the lowest levels of side effects, so like nausea and diarrhea, etc. And GLP-1 agonists on bariatric surgery or versus bariatric surgery. Can it, with all these consistent effects and with the most effective versions, can we maybe see it as a great replacement for bariatric surgery? So if you're interested, then join the Physionic Insiders and you'll get access to that as well. Okay, well, hopefully you learned something from this, and I'll hope to have the absolute pleasure of speaking with you in the near future. Bye.